Good morning, everyone. Welcome to today's FormuSense demo. My name is Monica Salib. I have a Bachelor's of Chemical Engineering, and I've been at FormuSense for about 6 years. Today's demo is pre-recorded. We should have about 5 to 10 minutes available at the end for live Q&A, at which point you are welcome to unmute yourself and speak up, or you can send your questions to the meeting host at any time through the WebEx chat. And you are, of course, welcome to contact me after the demo. There's no way to avoid the buzz around AI these days. It's being used for everything from search engines to self-driving cars, and of course, generative languages like ChatGPT. Over the last couple of years, we've noticed a real shift in the pharmaceutical industry, where AI and machine learning have become an integral part of this industry. We see companies forming their data science teams and investing in new technologies. In today's demo, we will show you some applications where FormuSense was used to predict titer and accelerate drug development. For more details on methodologies related to assembling an experimental dataset and building, interpreting, and validating predictive models, please consider attending our next 2-hour workshop being held on June 27th. Now, before we get further into our demo, let me tell you a little bit about FormuSense. We were founded by Dr. John McGregor, who is both a statistician and a chemical engineer. He spent his academic career as a professor of chemical engineering, pioneering the application of advanced analytics to industrial data. He used those 30-plus years of experience to go on to establish FormuSense in 2004. Now, almost 20 years later, we are proud to be carrying on his legacy and working with 3 distinct sources of data. Today, we will cover some of our offerings related to the use of experimental data as well as process analysis. But if you have any interest in machine vision, we would love to hear from you. FormuSense has extensive experience in the field of advanced analytics and product development. We've been applying our proven framework since 2004. As you can see, we have worked in numerous industries to help our clients achieve a variety of goals, from discovering new materials, reducing raw material costs, assessing sustainable alternatives, and mitigating supply chain issues, which was actually a hot topic over the last couple of years. So on that note, if you are not in the pharmaceutical industry, we're very happy to schedule a custom demo for you using a data set that you can relate to more. Using AI for product development isn't new for experts like FormuSense. However, it does still feel new to many manufacturers who are comfortable with conventional R&D workflows, like carefully planning single-factor DOEs that must be scheduled into a busy lab or pilot plant. In a conference that we attended a couple of years back, one of the pharma VPs told us that his data doubles every 5 months, yet his knowledge does not. That actually defeats the purpose of all of the PAT initiatives in place. As you are aware, the objective behind PAT is to have a consistent quality and an efficient process. And since PAT is a multivariate problem, there's no better way than applying multivariate statistics to it. FormuSense was carefully designed to tackle PAT corners when it comes to data collection and structure, model building, process understanding, etc. FormuSense can be an additional useful tool for you at different stages of development, whether you're looking at experimental data in early development, or large-scale pilot data, or actively working on improving the final production. Let me quickly introduce the software. Since incorporation in 2004, FormuSense has refined and applied our proven framework for data-driven product development and delivered that to our clients through consulting services. Over the last few years, our chemical and software engineers have worked very hard to streamline that product development workflow in an intuitive and no-code platform for our users, with the ultimate goal of reducing the need for physical experimentation. So what's that proven framework then? It essentially involves 4 steps. First, assembling your dataset to identify and resolve anomalies and ensure that it's suitable for advanced modeling. Second, building predictive models and interpreting the results with explainable AI. Third, applying that model in a forward direction to run digital simulations and predict quality. Then finally, inverting the model and performing numerical optimization in the backward direction to reach certain quality targets. We will briefly look at each of these 4 steps, but the primary focus of today's demo will be modeling and simulation. While each product development dataset and application varies somewhat, our approach is industry agnostic and is really suitable for both big and small datasets. and provides the flexibility to build models on the data that is available and relevant to you. All you need to get started is one block of input variables and one block of output variables. While there are endless applications for incorporating AI into the pharmaceutical industry, we wanted to showcase a couple of examples with ranging complexities. Starting with a simple example for titer prediction, this client didn't have any information about the formulations. All we had was the process conditions for 80 batches across 2 reactors and the corresponding final titer measurement that they wanted to maximize. The summary plots in FormuSense allowed us to easily observe the differences between the reactors, as you can see in the plot in the top right. The team was not aware that the reactors were ran differently, and that this is what led to inconsistent titer measurement. Using the coefficients and the contribution plots, the operational team was able to troubleshoot and pinpoint the key variables that led to a drop in titer, which mostly had to do with reactor A set points. This next example is more of a complex drug therapy development application, where our client was trying to formulate monoclonal antibodies that meet all the critical quality parameters at days 0, 1, and 3. Since we have the actual formulation available in this case, FormuSense generates a graphical summary on both the class level and the ingredient level. This is very helpful to give you a high-level overview of your historical formulations and usage of the ingredients. Looking to the right, we see a bar plot that summarizes the class usage. For example, amino acids were used in 100% of the formulations with a ranging percentage between 24 and 45. Comparing that to nucleosides, they were only used in about 80% of the formulations. Also, similar to the previous example, the summary score plot at the bottom is colored by 2 different cell lines. The clustering indicates that data was significantly different between these 2 cell lines, and a submodel on each of them would be more appropriate for further investigations. Once we built a submodel on each cell line, we were able to unlock the full potential of the model's predictive ability. Coefficients can now be explored for each of the critical quality parameters. For example, a higher glucose on day 3 was correlated to a higher concentration, whereas formulations with no nucleosides had a lower TCD on day 3. These findings can often direct the development efforts to an optimized monoclonal antibodies in a much faster approach than standard DOEs. This final example showcases the full framework of FormuSense. Our client was interested in modeling the impact of mixing different pastes on the final bulk quality from 2 different lines. In this case, we're no longer restricted to formulation ratios and process conditions only, but we also have information about the properties of the paste. This is the most powerful model structure, since it allows us to consider new ingredients and new mixtures that were never tested before, given that their properties were actually measured. Let's take a look at this data set in FormuSense together. The first step is to import the data. Once I click Import, I can go and get my Excel file and then specify each of the tabs. So my first one here is Formulations, the second one is my Ingredient Properties, the Paste Properties. These 2 are the Process Conditions, and finally I have the Quality. Once this is all set, I can click OK. In the Formulations tab, I need to specify what's the name, the ingredients, the quantity, the class names, and so on. Once all of that is set, I can now click OK. I get a nice summary here on the top right, just about the data set, how many formulations I imported, how many ingredients, how many classes. You can click each of your formulations here to view them again and just confirm that everything is right. You can navigate through the different sheets of data that you imported. So I have the ingredient properties, and you can just keep scrolling through your ingredient properties to make sure you don't have any data integrity that you need to correct. If there is any need to fix any data anomalies, there's some options that you have here that we're going to cover in more details in the workshop, like replacement rules and data fixing and so on. I'm going to skip over the ratios part here because we previously looked at the class usage and ingredient usage bar plots together in the previous example. More importantly, I just want to highlight that feature of having mixture properties automatically configured and generated in FormuSense. Since we have all properties measured on all pastes,
we can actually go ahead and generate global mixture properties. That allows us to consider new pastes that have never been tested before in future simulation and optimizations. All you have to do is click Generate Mixture Properties, and now we get that final mixture paste properties for each of our formulations.
Let me give you a little bit more details about what happens in the background of mixture properties configuration. We use applicable mixing rules to combine the formulation ratios, which we typically refer to as R, and the ingredient properties, which we refer to as X, to form that new mixture properties block Rx. These two blocks of data are combined using linear mixing rules by default, but FormuSense has some additional options to account for non-linear mixing rules if you'd like. Following the example here, if your formulation has 40% paste A and 60% base B, then the molecular weight of the mixture would just be a weighted average of these two.
Jumping ahead to the modeling part, once we generate a model, we get three distinct views: a summary view, a pruning view, and an interpretation view. For the sake of time, I'm just going to spend most of the demo today highlighting the interpretation features here. This is where you get the observed versus predicted plot for your quality variables and the corresponding coefficients that explain what key X variables are highly correlated to that quality of interest.
By looking at the coefficient plot here, you can see that we have three different colors, and that corresponds to the input X blocks that I've imported into my model building step. I have process 1 in purple, process 2 in yellow, and the generated paste global properties in green. Just by looking at the coefficients, you can see that the yellow bar has the highest impact on the quality, which is the process conditions coming from Unit 2. If I select this bar, the raw data is automatically going to be viewed on the right here. And if you would like to confirm these findings, I can select these low points here, and they will be automatically selected on the quality variables as well. Since this is a positive correlation, it makes sense that the lower that variable is, the lower the quality variable is. The real benefit of these AI predictive models is all about using them in the forward and backward directions interchangeably. Pilot plant trials can be effectively reduced by using the simulation feature to predict the outcome of a specific formulation without actually running it. Also, numerical optimization can be performed to back-calculate the formulation and process conditions required to reach the desired quality outcomes. Let me show you how easy it is and user-friendly to run different simulations in FormuSense. All you need to do is go to the Simulations tab here under the Setup and configure your different scenarios. For example, I started by scenario 1, which is exactly the process conditions of observation or formulation 176, and I picked two different pastes, paste 1 and paste 100, 50-50 ratio. And then I ran a different simulations by changing the paste, so instead of paste 100, I now picked paste 134. All you need to do is just check the paste that you'd like to consider and add its percentage. My third scenario is changing the paste percentage. So instead of having a 50-50, I now have an 80-20. And then finally, for the fourth scenario, I changed some conditions in process line 2. If you remember, that was one of our highest coefficients in the model building step. Once you're done configuring your scenarios, you can go ahead and generate simulations. You can see where your four scenarios are projected in the score plot here. You can see the Y predicted for each of these scenarios, where 0 here means the average, and the number here is the standard deviation higher than average. So you can see that my final scenario, where I changed the line, has the highest impact on my quality variable. You can also check the formulation, you can check your process conditions, your mixture properties, and there's a very nice feature called Find Closest. Because what's the point of running an experiment if you had run something completely close to it in the past? So once you click Find, for each of your scenarios, you will get the closest matching observation that you had previously run before. To conclude, I hope I've shown you the value of predictive modeling for product development applications in the pharmaceutical industry. I would encourage you to take advantage of the 30-day trials that are publicly available on our website. Just note that the optimization, which again is still in development, will not be included in the trials. Please reach out if you have any questions about licensing or consulting services. If you would like to take a deep dive on the methodologies behind our framework, please consider joining our 2-hour workshop that's coming up by scanning the QR code or simply visiting our website. Thank you very much.